So, have you ever wondered how drug companies make sure that every single pill in a bottle works exactly the same way, especially when they have to make tiny little changes to the recipe? Well, today we're diving into a really brilliant piece of science that's like a shortcut, connecting what happens in a lab beaker to what actually happens inside your body. Let's pull back the curtain on this.
Okay, just imagine you're a drug manufacturer for a second. Millions of people are relying on your medication, right? But then you need to switch suppliers for some minor ingredient. Or maybe you move your whole production to a new factory. How do you prove the medicine still works exactly the same? I mean, do you have to run a brand new, super expensive human trial for every single tiny tweak? It's a huge problem, especially for one particular type of drug.
And yeah, that problem gets especially tough when we're talking about extended-release drugs. With these, the stakes are just incredibly high.
You see, extended-release or ER drugs, they're not like a normal painkiller that just hits you all at once. Nope. These things are little marvels of engineering. They're designed to dissolve slowly, releasing the medicine into your system over, say, 8, 12, or even 24 hours. For anyone who needs steady medication levels, that timing isn't just important, it's absolutely critical. So, back to our manufacturer. What happens when they need to make one of those little changes we were talking about? A tiny adjustment to the pill's coating, a new piece of equipment, you name it. How can they be 100% certain that this new version will still dissolve at the exact same rate inside a person's body? Because running a full clinical trial for every minor change is just way too slow, way too expensive, and honestly, pretty impractical. There just had to be a better way.
And guess what? There is a better way. It's a kind of scientific bridge, one that's designed to connect the simple, controlled world of a laboratory to the complex, kind of unpredictable world of the human body. To really get this, we need to know two key terms. First up is in vitro, which is just Latin for in glass. This is all the stuff you do in a controlled lab, like dissolving a pill in a beaker of fluid. It's simple, it's clean, it's repeatable. Then on the other side, you have in vivo, which means in the living, and that refers to all the incredibly complex stuff happening inside a person. The whole goal here is to make that simple in vitro test reliably predict the complex in vivo result.
And this brings us to the hero of our story, In Vitro/In Vivo Correlation, or IVIVC for short. Basically, it's a predictive mathematical model that creates a solid, scientifically proven link between those two worlds. If you have a strong IVIVC, you can take the data from a simple lab test and confidently predict how that drug is actually going to be absorbed by a patient. It's a total game changer.
But this confidence didn't just pop up overnight. What's really wild is how quickly the thinking on this changed. Back in 1987, the scientific community was pretty skeptical. They thought a reliable correlation just wasn't possible. But then, only 3 years later, the whole mindset shifted. It suddenly became a really important goal. And all that work led to 1997, when the FDA released its official guidance, which basically created a standard playbook for how to do this. Now, as you can probably guess, not all these predictive models are the same. To be really useful, they have to be graded on how accurate they are. So let's take a look at how that's done.
Right at the top, you have the absolute best of the best, the one everyone is aiming for. It's called a level A correlation.
This is the gold standard because it creates a direct point-for-point relationship. So what that means is, if 10% of the drug has dissolved in the lab after 1 hour, you can predict exactly how much has been absorbed in the body at that same time. It lets you map out the entire journey. The best way to think about it is like having a GPS with live traffic updates for the drug's path through your body. You know the whole route, the speed, and the timing, from start to finish.
But, that kind of detail isn't always needed. Sometimes a simpler picture is all you need, and that's where something called a level C correlation comes into play.
So instead of mapping that whole journey, a level C correlation is more like a single snapshot. It connects one specific lab measurement, like how much is dissolved after 4 hours, to a single key result in the body, like the peak concentration of the drug in the bloodstream. Using our travel analogy, this is like knowing your final arrival time, your ETA, but having zero information about the route you actually took to get there. It's useful, for sure, but it doesn't give you the whole story.
And that's exactly why the FDA, in that big 1997 guidance, made it crystal clear what they prefer. They said, and I quote, "A level A IVIVC is most informative and is recommended if possible." In other words, they want the full GPS, not just the ETA.
So, why on earth would you go to all the trouble of developing this really sophisticated level A model?
Well, here it is. This is the big payoff. This is the ultimate reason IIVIC is so important. It's a shortcut.
Once a company has a validated, predictive Level A IVIVC, they can apply for something called a biowaiver. And it is pretty much what it sounds like. The FDA officially waives the requirement for them to do a new human study after they make certain changes. The lab test literally becomes the stand-in for the human trial.
And this is the real game changer. With that strong IVIVC, a company can make a small change to an inactive ingredient, they can update a process, or even move production to a whole new factory. And as long as the new pills perform identically in that lab test, they can skip the super expensive and time-consuming human study. It's an amazing way to ensure safety and consistency while also speeding up innovation.
So let's bring this all home and look at the actual real-world impact that this guidance document had back in 1997.
The data really tells the story here. In the decade before the guidance came out, when confidence was still pretty low, only about 15% of extended-release drug applications sent to the FDA even tried to do an IVIVC. But in just the 2 years leading up to the official guidance, once the science was accepted, that number shot up to 75%. The industry was clearly ready and waiting for this.
And that's really the bottom line. The development of IVIVC completely transformed a routine lab test, the dissolution test, from just a simple pass-fail quality check into this incredibly powerful predictive tool. It became a window that lets us see how a medicine will actually perform inside a person, making our drugs safer, more consistent, and a whole lot easier to improve.
It really is a remarkable achievement in science. And it kind of leaves us with a pretty fascinating question to think about. As our technology and our understanding of the human body just keep getting better, what's going to be the next great scientific shortcut that helps us make medicines even safer and get them to patients even faster?